This lesson, we're going to look at sexual reproduction and variation, and we're going to look at the stages of meiosis. And not to be mistaken with mitosis. However, there are some similarities between meiosis and mitosis. My advice to you is watch the video on mitosis if you're not aware of the process of mitosis in cells, because there are some similarities, as we're going to use the same phase names during this concept of meiosis as we did in mitosis. Now, for sexual reproduction, it's a reproductive process that involves two sexes, right? And so we have genetic material that is shared between one parent and another. So they combine together to form offspring that have some genetic similarities from both parents, right? Think about yourself, right? If you know, as a young adult or as a young child, and someone says, oh, you know, you've got your mom's eyes or your dad's nose, maybe you have, you know, your mom's hair color or your dad's hair color, right? So this, right, this notion of I've got this from one parent, this other from another parent, is really that concept of what we call variation. Right? And how do we bring together genetic material from two individuals to form a completely different offspring? Right? Now, in animals, animals reproduce sexually, right? And so they produce gametes, right? They produce gametes, right, in reproductive organs called gonads. Right? Now, in males, right? In males, right? The gonads are called testes, and in females, the gonads are called ovaries. Now, in order for a human chromosome number, right, to remain 46, right? Remember, well, you should know that humans contain 46 chromosomes, right? So you as an individual have 46 chromosomes. 23 of them, right? 23 of them came from a male. The other 23 came from a female, and they combine together to form 46 chromosomes. However, in human chromosome number to remain as this 46 chromosomes, right, gametes must have half the number, right? So these gametes somehow, right, think of every cell in your body, right, as humans have 46 chromosomes, with the exception of these of the human sex cells, right? So testes form sperm, ovaries form eggs. So these are the only cells in the human body that actually have only half the number of chromosomes, right? So half the number of chromosomes, right, of 23, is referred to as haploid. So we could say that sperm and egg cells, right, have a haploid number of chromosomes, which haploid meaning half, right? So only haploid gametes, right? This sperm, right? This egg, these are gametes, right? These are gametes. And these gametes have only half the number of chromosomes. Now, during fertilization, oh my goodness, I don't know what I'm saying here. During fertilization, we're going to have a sperm that is going to come together with an egg, right? And they're going to fertilize. So what they're doing at the time of fertilization, they are combining the haploid number of chromosomes together to form this zygote, right, which is really the very first cell that made us who we are today, right? We started off as with an egg and a sperm that come together, right? If you're one of my viewers, odds are you're a human viewer. That sperm, that egg came together to form this zygote that contains 46 chromosomes, or as we can say, a diploid number of chromosomes. So the gametes, sperm and egg, have haploid number of chromosomes, while the zygote in every other human cell contain a diploid number, which is double that number of the haploid cell. Now, how does this happen? Well, this really starts from the 46 chromosomes. So as we said, these 46 chromosomes have to find a way to divide that diploid number into haploid numbers. And this process that this happens is called meiosis. And meiosis is the process that ensures that the gametes, right, contain, right, that these gametes contain only half the number of chromosomes as every other cell in your body, right? Half the number of chromosomes. Right? Now, also, meiosis also ensures that the gametes have a different combination of chromosomes, right? So your body has specialized organs that help make these gametes for sexual reproduction, right? As we said, the gametes are either sperm or egg cells, right? So the gametes from two parents combine, right, in a process, as we said, called fertilization, that forms this zygote. And this zygote is what's going to ultimately undergo the process of mitosis that I've mentioned in my previous video. All right? So, as we said, humans, right, contain 46 chromosomes, 23 pairs, right? So let's look at this number here now. Humans contain 46 chromosomes. Right. As we said, 23 came from the male, 23 came from the female. So in your 46 chromosomes, so in your cell, right? So you got 46 chromosomes, right? So let's try to draw something here. So let's try to draw that cell. So here's our cell, and in that cell, and I'm not going to draw it like I've drawn it before, in that cell we've got 46 chromosomes. So what I'm going to do is I'm going to draw the chromosomes like this. And what we have here is 23 pairs, right? So we've got, here's one pair, so that's 2 chromosomes. And we're going to go down to our last pair, right, which is the 46th set of chromosomes, 45th and 46th set of chromosomes, or should I say the 23rd pair. So here is the first pair, and these dots represent all the other sets, and here we have what we call our 23rd pair. Now remember, each pair has 2 sets of chromosomes. One that came from one parent, the other came from another. Right? So let's look at what have 46 chromosomes. So body cells contain diploid number, right? Diploid number of cells, right? That's 46. Gametes, right, or the sex cells, contain that haploid number of 23 chromosomes. So now, what ultimately happens? So what we're going to look at is the and for this to happen, right? These cells, right? These sex cells are ultimately going to be formed through a process called meiosis. And we're going to look at meiosis a little bit closely. So, first stage of meiosis, and I'm going to simplify this, is referred to as prophase 1. And in fact, going back to meiosis, right, one thing I ended up forgetting to state is that meiosis actually comes in 2 phases, right? And those 2 phases are meiosis 1 and meiosis 2. And you're going to understand why we have the 2 phases as I continue to draw this. So in prophase 1, right, this pair, and actually another thing that I forgot, sorry, right, if we're going to look, I'm going to highlight here this pair, this first pair. As we said, this first pair, as we said, one came from one parent, one came from another, are referred to, right, as homologous chromosomes. Homologous chromosomes mean that they are similar chromosomes to one another. What do they mean by similar? Well, the location of this centromere is exactly the same in both, right? One came from one parent, one came from another. Some of the genetic material that is found on these is exactly identical. So if we look at in a little closer, let's say on this part of the chromosome and this part of the chromosome, let's say that part of the chromosome refers to hair color, right? So it's found obviously on both sides, right? So hair color chromosome or the code for hair color is found at the top part of this, let's say, chromosome from one parent, which is found on the same part of the other parent. So they pair up according to what kind of information that these chromosomes will give off. And that's what we refer to as this homologous chromosomes. And in prophase 1, these homologous chromosomes pair up with one another. Right? So they find each other and they pair up. Right? So we've got here from one parent, here from another. And watch what's going to happen here. We're going to see this here sometimes. This is going to happen sometimes. We're going to have ends of the chromosome that are going to cross over. And we're going to talk about crossing over in just a moment. So crossing over of chromosomes. So we've got a pair here of homologous chromosomes. We've got another pair here of homologous chromosomes, right? And remember, one, and we're going to cross over here again. And so they're paired up during prophase 1. So after prophase 1, the cells undergo the next phase, which is called Metaphase 1. And Metaphase 1, very similar to Metaphase in mitosis, but the only difference is as follows. So here what's going to happen is we've got our cell, and very similarly to Metaphase in mitosis, Metaphase in meiosis, the chromosomes still line up along the center of the cell. However, they don't line up in line as they do in mitosis. They line up as their homologous pair. Right? So here, notice they're lining up along this center here, right, as pairs. And as we said, on one side of the pair is from one parent, the other side is from another parent. So we have this crossing over, right? And this crossing over is what gives rise to this term here that I mentioned here at the beginning called variation. You're going to see how this crossing over is what allows for mixing of the genetic material, right? And you're gonna understand that mixing in just a moment as we move from metaphase down to the next phase called, and hopefully you guessed it, anaphase. And it's anaphase 1, right? Because we're in the first stage of meiosis. And what happens here in this phase of anaphase is that the spindle fibers are going to start to pull apart these homologous pairs of chromosomes, right? So, and watch what happens here. Because of this crossing over, and this is really where this notion of crossing over is going to play. And if you notice here, really what's happening is the following. So notice how, let's say, the blue chromosome and the fully red chromosome now, when they are being pulled apart, are carrying some genetic material. So let's say, as we said, this red chromosome came from a female donor. Now this chromosome has a part of the male donor with it, right? And the same thing will apply to the bottom pair of homologous chromosomes, right, with itself. So here we go, and please bear with me having to draw this, but this is the only way to help really better understand this concept of crossing over and meiosis. So ultimately, and really what we have, we've got spindle fibers that kind of pull them apart, right? And after anaphase 1, right, so now we've got the homologous pairs. They are no longer going to be paired up. They're being pulled apart, right? So we've got, now let's look at the count now. Going back, we've got 46 chromosomes. We still have 46 chromosomes. We're going to move now into the next phase, which, if you guessed telophase 1, you guessed correctly. So we are into telophase 1, and in telophase 1, we've actually got 2 cells, right? But they have now the chromosomes, right? They don't have the homologous pair anymore. They actually have the haploid number. So now we've divided the 46 chromosomes that we've started into 2 cells that have 23 chromosomes. And let's draw the other. Right? And we have here on this side. So notice which chromosome we've got on each side. Right? We've got half, right, of the homologous pairs. Right? We've got so. And let's continue with this drawing here to show. So now notice here we've got a part of the genetic material, and notice how both of these cells are now starting to look completely different. By the end of telophase, during mitosis, both of these cells are exactly identical. But at the end of telophase 1, these cells are going to be completely different. And now these 2 cells are going to undergo the second stage of mitosis, or of meiosis, which is referred to as meiosis 2, because we're not ready. This isn't enough, right? This isn't enough just yet. So, because remember what we said in the cells, right? Remember what we said about the cells? The cells, as I've said in during the mitosis video, are as chromatin, right? They're single strand. These are double-stranded. So somehow we need to undergo meiosis 2 that will form these as single-stranded chromatin, right? So telophase 1 is going to now enter into meiosis 2. And more importantly, we are going to be entering into what we call prophase 2, in which in prophase 2 now we've got the double-stranded, and it's not really much different, right? The double-stranded, right? The cells are now going to be getting very close to pretty much lining up along the center. So there really isn't much of a difference. Now, let me draw that blue piece there. Put in the red. Right. So here's the other chromosome. So telophase 1 and prophase 2, not really much different. We're not going to look at it as being much different, apart from the fact that telophase 2 it really is just where the cell actually divides itself. Right. So it's kind of splitting. Well, the truth of the matter is really that they're still kind of connected, and then once they split up, that's really where prophase 2 really lies. Right? So here we have now our final chromosome, right, with its piece of green genetic material. So we've got prophase 2, and that cell is now is getting ready to move into metaphase 2. And in metaphase 2, again, very similar to metaphase and in fact, meiosis 2 is very similar to mitosis itself, because what's happening now is the chromosomes, or the haploid number of chromosomes, actually line up along the center of the cell. Right, they line up along the center of the cell. All right, and they line up along the center of the cell, where the spindle fibers will be ready to start to pull these chromosomes apart. Right. So after metaphase 2, right, you can almost figure out what the next phase is, and that's Anaphase 2. And now, make note of what's actually going to happen with these two cells. So what's going to happen is now these spindle fibers are going to pull apart at these chromosomes, and they're going to form half. All right. So what we've got here, oops, sorry, I know it seems it's taken a while, but it's the only way to really understand meiosis. Just looking at the pictures as is, and just seeing the thought process that goes on is what I find just valuable. So what happens is these chromosomes get pulled apart on either end, right? And so here we have now the green one, and with part of the green one, right, we've got the purple one. Let's put the green one on this side here. So we've got the purple being pulled apart, and then the full purple chromosome on this side. So notice now, all of a sudden, again, our count, right, if we're looking at it, right, all these are still 23, right? So they're all the haploid number, 23. Now, after Anaphase 2, what happens is, you guessed it, we're going into Chilophase 2. And in Chilophase 2, watch what ultimately happens. This cell is going to split and form four, right, sister cells, or right, and these are all going to be different. So what's going to happen is in one, right, we've got the blue, we've got the green genetic material, but in this one here, this second one, right, because what's happening is this cell here divides into these two. This cell here divides into these two. And watch what happens here, what they actually will look like, right? Note the difference in the genetic material that we have, right? Note just how different, right, these cells are to one another, right? And notice that variation that we were talking about, right? And this is how genetic material changes over the years. And if you look back at even ancestry, and you go, well, yeah, you may look at grandparent pictures and go, oh my goodness, yeah, you could see how one parent looked the same as your grandparents, right? Because this genetic material, who knows what was passed over the years. Now notice here we've got four completely different combinations. And what's going to happen is these four are either going to form either sperm cells, or they're going to form egg cells. And there's a little difference. So imagine this one cell, when it forms the sperm at the end, right? Notice the genetic material. It's containing genetic material from two parents. Who knows what combination lies at the very end when they get formed, right? Remember, this is occurring in 23 pairs of chromosomes that are pairing up, right? These, remember we said here towards the beginning when we were talking about the first pair and the 23rd pair. Look at how the possibilities, imagine if all 23 pairs crossed over, right? If all 23 pairs underwent this crossing over, think about the possibilities of the different types of sperm cells that we can actually form, right? So here's the thing though, right? When we undergo spermatogenesis, right? When we undergo spermatogenesis, right? And that's formation of sperm, right? We have the possibility of forming four completely different sperm cells by the end of meiosis, right? So imagine, right, four different sperm cells all having slightly different genetic material, but they all came from very similar genetic material. But this is the way we have variation. So all four will form completely different cells, even though every single cell in our body is exactly the same. However, when we form the sperm, that's where the variation lies. Now, the difference though during egg cells, and the formation of egg cells is what's referred to as oogenesis. Right? So we're forming egg cells. However, the difference here is only one of these, right? So if we look at this formation, right? If we're looking at this formation, in fact, what happens in females is at the end of meiosis 2, right? At the end of meiosis 2, only one of these is going to survive, right? And the rest, the material is just going to disintegrate. And then, so we're not during, if we undergo prophase 2, we're not going to have this part happening, right? So we're not going to have this. However, when that one haploid cell undergoes meiosis 2, and this is only in females to form the egg, right? This cell, when it undergoes prophase 2, metaphase 2, anaphase 2, and then finally telophase 2, only one of these is going to survive. So at the end, in female cells, right, and for the egg cells, right, for the egg cell, only one egg gets formed, while by the end of meiosis 2, there are 4 possible sperm, 4 possible sperms with all completely different genetic material. But look at what kind of genetic material. It all depends on what kind of characteristics get passed on. You could have complete variation, or you might have many common similarities, where you might see, you know, a daughter looking almost identical to the mother, let's say, depending on what part of genetic material. And again, then that goes into dominant and recessive genes, but we're not going to worry too much about that. So that, there you have it. That's pretty much what occurs. So in males, meiosis actually occurs continuously in the testes once the gonads are, right? So once a male undergoes puberty, pretty much this, for the, you know, the formation of these gametes is when it occurs. But now in females, meiosis begins in the ovaries before birth, right? And then it stops at about 12 years of age, 13, 14, when the female finally starts to undergo puberty. So females form all their eggs then, right? And they stop by the time they hit puberty, while males will continuously form, you know, will continuously undergo meiosis.